Hi, I'm Sadashiva Pai from Science Mission. Today we have Dr. Fabrisia Ambrosio, Associate Professor at the University of Pittsburgh Medical Center is with us. She has published several research papers to her credit, and she has won several awards. I'll just name a few of them. In 2018, she was University of Pittsburgh Institute of on Aging Best Junior Faculty Research Award. In 2013, presentation at the PT in Motion magazine featured investigator of the cover article, A Growing Concern. Again in 2013, Regenerative Medicine, a key to tomorrow's rehabilitation. In 2013, Drexel University Alumna Profile, Dr. Fabrisia Ambrosio combines passions for physical therapy and regeneration medicine. In 2014, PT in Motion magazine featured investigator. In 2015, Carnegie Science Center Award. She was awarded NIH Top Paper of 2016. Awarded Aging Cells Best Paper of 2017. And in addition, she has received several research grants. Welcome, Dr. Ambrosio, to Science Hangout. Can you please talk about your background, education, and all?
Sure. So I was born in São Paulo, Brazil. I came to the U.S. and I've done my training here in the U.S.
Okay.
My baccalaureate training was at Mansfield University, where I majored in biology with a minor in chemistry. When I finished my baccalaureate, my two main interests were really my favorite courses during my baccalaureate training were physics and biology. My father was my physics professor in undergrad, so I was interested in kind of pursuing some of those topics. I got an interest in rehabilitation and applied to physical therapy school. And I also applied to do a Master of Science. Laval University. So I was accepted into both of those programs that I had applied. And so it was a difficult decision. What I decided to do was defer my acceptance at the physical therapy school at Drexel University in Philadelphia to go up to Quebec, where I liked the idea of kind of immersing myself in French. It was a language that I wasn't too familiar with, so I thought it would be a great experience. And so I was up in Quebec for about a year, and then I went to Philadelphia, and in that second year, I actually did both programs concurrently. So I was, although neither program knew it at the time, but so then I finished with my training in Drexel University in physical therapy, and I already was kind of committed to a field in research. So I ended up applying for a PhD program in Pittsburgh, at the University of Pittsburgh, in the Human Engineering Research Laboratories. And most of my work there was clinical research. But then when I got my faculty position, I decided to do more basic science translational work.
Okay. And can you please tell me about your lab people, grad student, postdocs?
So we have a nice mixture of trainees in the laboratory. I have doctoral students from bioengineering and also from environmental health and occupational safety. Some from the Department of Medicine, a postdoctoral fellow. So we're pretty diverse in our interests, which I think makes for very fun and lively lab meetings each week.
Okay. And how do you motivate them?
So one of the things that we recently started in the laboratory is we have our weekly lab meetings, but we decided to launch what I'm calling a Innovation Heroes series. And essentially our idea is to kind of present, very briefly, 10, 15 minutes, an innovation hero, somebody who's kind of taken chances, stepped outside the norm, and we kind of present this hero, failures that they've encountered, and, kind of, what does that innovation hero profile look like, and what does it take to really be a creative mind? And so it's been a lot of fun. We've had, I presented on Richard Feynman, who is one of my own personal innovation heroes, but we've had presentations on, for example, Freddie Mercury was a recent one. So it's a fun way to kind of look at the profile of a successful and innovative individual. And it's also a great opportunity to kind of learn more about some of the personal interests of the people in the lab.
Okay, and you mentioned about your interaction with the lab people in terms of weekly meeting and all. Anything else you do to help them out?
So, of course, we have a bunch of informal meetings, but the other thing we do once a week, we have sort of mini meetings where we get individuals who are kind of working together on papers. And so we have kind of paper-focused meetings where we kind of talk about the data, you know, figure 1, figure 2, map it out so that we can really be focused on the story as a whole, where we need to go. And, of course, we take the opportunity to also discuss, you know, challenges that we've encountered. So that's been something that we've been doing. I found to be pretty useful.
And let's talk about your research, like your work on Clotho and all those things. If you can present your work, that will be great.
Sure. I'm going to just do the screen share then.
Okay.
Okay.
Here you go.
Is it showing?
Yep.
Okay. Yeah, so my laboratory is primarily focused on skeletal muscle regeneration, cascade, and muscle stem cells. And in this particular project, we're very interested in understanding some of the molecular and cellular mechanisms underlying an age-associated reduction in skeletal muscle healing capacity. So I like to start with this image because I think it really shows the importance of maintaining, you know, high levels of skeletal muscle functioning over time and how much of an impact it can make, not only on physical mobility, but also on quality of life. So actually we have learned a lot. You know, the field has really learned a lot about the skeletal muscle regenerative cascade in the last, you know, almost 60 years since the muscle stem cell was first discovered by Alexander Mauro. So just a brief recap of what we know about the skeletal muscle regenerative cascade, because skeletal muscle really has a great capacity to heal itself after an injury event. So muscle fibers contain a very small population of muscle stem or satellite cells. Now, they're called satellite cells because they have a very precise anatomic location, and that is here between the basal lamina and the plasma membrane. And these satellite cells reside in a normally quiescent state. That is, until a muscle injury or some type of physiological stress actually calls these normally quiescent satellite cells into activation. So once activated, the muscle stem cells will move to a state of proliferative expansion, really to amplify the stem cell numbers at that injury site. These activated stem cells will fuse together and they form a actively regenerating muscle fiber that is characterized by, as you can see here, this central nucleation. And the central nucleation turns out to be a really nice tool for us to be able to go in and histologically take a look at, you know, what are the myofibers that are regenerating versus those that are, for example, damaged or even mature. Of these actively regenerating muscle fibers, though, what we know is with time, the nuclei will migrate back out to the periphery, and we see this great restoration of the quiescent satellite cells. And so I think the key thing is that, you know, when this process goes well, as is the case of young, healthy skeletal muscle, what we see is a very nice restoration of the original structure and function of the damaged muscle fiber. However, what we know is with aging, that regenerative cascade becomes markedly impaired. So these are just histological sections of cross-sections of young injured skeletal muscle and old injured skeletal muscle. As you can see here, that central nucleation that I mentioned, very prominent with these nice, you know, larger fiber diameter myofibers in the young injured, but what is greatly compromised in aged skeletal muscle. And so we see that the myofiber diameter is much, much smaller. And after time, what we see is that functional muscle fiber regeneration is impaired and really characterized by an increase in fibrosis at that injury site. Of course, the deposition of fibrosis is important because fibrosis, not being contractile in nature, will dramatically affect the force recovery of that muscle. And so here, this is some in situ contractile testing that we performed in young and injured muscle 14 days after injury. And what you can see is that the force recovery of that old muscle is significantly lower than the young counterparts. Now, there's been some great studies that have really looked at the cellular and molecular level to try and understand this decreased myofiber regeneration and increased fibrosis that we see with age. And perhaps not surprisingly, what Brock et al. Demonstrated now over 10 years ago was that with aging, we see a predisposition of the muscle stem cells to differentiate toward a fibroblastic lineage. And so this so-called myogenic to fibrogenic conversion is believed to contribute to some of that increased scar tissue deposition that we see in aged muscle. Interestingly, that myogenic to fibrogenic conversion has been associated with and linked to an increase in Wnt signaling activation. But there's good news, and in that same paper by Brock et al., they demonstrated that these age-related changes are indeed reversible. And so they did this using a very elegant model of heterochronic parabiosis, where essentially a young mouse is sutured together with an old mouse, such that there's an exchange of the circulating factors between the two animals, essentially creating a chimerism. And in this paper, what they found is that exposure of aged mice to youthful circulating factors significantly enhances the regenerative cascade, in large part due to an inhibition of Wnt signaling activation. So these exciting experiments and findings have really triggered the field to take an interest in what could potentially be the circulating factors that might be contributing to this enhanced regeneration in old mice when exposed to youthful blood. An important aspect of this study was that they demonstrated that it wasn't stem cells from the young mouse that were being transferred into the old mouse. And so microenvironmental alterations owing to this youthful blood in the aged mouse seem to be really driving an enhanced regenerative potential of the endogenous stem cells within the old animal. So we became also, after these studies, we also became very intrigued as to what might be some of the circulating factors playing a role in this rejuvenating effect of young blood. And so we turned our attention to one protein, a so-called longevity protein, Clotho. And we wondered if this could be potentially a contributor to some of the anti-aging effects of a youthful circulatory system. We turned our attention to Clotho because it was previously shown, in fact, it was the same issue of Science in 2007 that demonstrated that Clotho modulates stem cell functioning. And not only that, but it actually does so through inhibition of Wnt signaling activation. And the same paper also implicated Clotho as playing an important role in tissue regeneration. And so we wondered if there may be a role for Clotho in the skeletal muscle regenerative cascade. So when we started to look a little bit more into Clotho and try and understand some of the background, we found it to be a very fascinating protein. So very briefly, Clotho was first discovered back in 1997 by Kuro et al. And essentially, it was a serendipitous, what they call a serendipitous discovery, where a mutation in what they now know to be the Clotho gene led to a rapidly accelerated aging phenotype in the mice. And so here you can see the wild type versus the aged Clotho hypomorph. And whereas a wild type mouse will typically live about 3 years, In the absence of a functional Clotho gene, they found that these mice lived approximately 2 months. And over this time, they developed just a myriad of aging phenotypes, including cardiovascular dysfunction, cognitive dysfunction, as well as sarcopenia. Now, I think what was very remarkable is that subsequent studies demonstrated that overexpression of the Clotho gene led to an enhanced both lifespan and health span. Now, subsequent epidemiological studies have also demonstrated that Clotho is important in humans. And it's been suggested that Clotho steadily decreases in the bloodstream of individuals over time. And epidemiological studies have shown, much as was shown in the mice, that lower Clotho levels are associated with cardiac dysfunction, cognitive impairment, and what they show here, maintenance of skeletal muscle health. And so this was a paper that was published by Semba et al. In 2016. And essentially, they stratified individuals according to their levels of Clotho expression within the blood. So they had low Clotho, medium, and high Clotho levels. What was fascinating is when they followed these individuals for several years, they found that those individuals with high Clotho levels in the bloodstream actually displayed a preservation of skeletal muscle strength, or a decreased loss of skeletal muscle strength over time, implicating Clotho as potentially playing a role in the preservation of skeletal muscle health and vitality. So we decided to take a look at the role of Clotho in skeletal muscle regenerative cascade. So we started off just looking at healthy, uninjured, young, and aged skeletal muscle. And in fact, this is just a cross-section of muscle from the two groups. Green here is in Clotho, and as you may be able to appreciate, we see very little Clotho expression in muscle under conditions of homeostasis. However, what we found is that in young muscle following an acute injury, and this is 14 days after injury, we see a very robust Clotho expression at that injury site. However, in aged muscle, we saw that Clotho expression was markedly attenuated, even following an acute injury event. And so that is quantified here in the graph on the right side. We also found that the increase in local Clotho expression at the injury site of young muscle was associated with a significant increase in Clotho within the serum. But just as we had seen locally in aged muscle, we saw no increase in circulating Clotho levels following an injury event. And this is just PCR looking at young versus aged skeletal muscle at various time points after injury. So we have a baseline, 3 days after injury, 7 days after injury, and 14 days of injury. And just as we saw at the protein level, we see a dramatic increase in Clotho gene expression 3 and 7 days after injury, but that change in expression is blunted in the aged counterparts. Of course, those studies are very correlative, where we see a blunted Clotho response in aged mice. And so to begin to explore whether Clotho may be having a more direct role for skeletal muscle regeneration, we turn to mice that were engineered to be heterozygously deficient for Clotho expression. And when we looked at the regenerative cascade in these mice, indeed, we found not only a significantly decreased Clotho expression at that injury site, as was expected, but we also saw that this was associated with a decrease in the regeneration index, or the number of centrally nucleated actively regenerating fibers, as well as a significant decrease in the fiber cross-sectional area. And these findings were consistent with previous reports of an impaired regenerative response in Clotho hets by Ahrens et al., which is actually published earlier this year. We then wanted to manipulate Clotho more locally within our animal model. And so using an shRNA inhibition approach to Clotho, we inhibited local Clotho expression in young mice. And this is just kind of validating that indeed we see a significantly decreased Clotho expression within the young muscle after injury. And when we did this, then we wanted to take a look at whether gene silencing of Clotho in our young mice might impair the regenerative response, similar to what we had seen in the Clotho hets. And so here on the left, we have second harmonic generation images, which are just essentially 3D constructs of the myofibers in green and collagen fibrils in red. And so in these movies, what we can see is young, healthy mice 2 weeks after injury actually display a very nice regenerative response, as would be expected. So after injury, we see very nice myofiber architecture. At times, I think you may be able to appreciate the myofibers, which show a very robust central nucleation. The myofibers are very dense. And again, the organization is pretty nice. Here we have a cross-section, again, allowing us to see that muscle organization as well as the central nucleation of the muscle fibers. Now with these 3D constructs, then we can extract out the myofiber channel and just take a look at the collagen. And essentially what we see is a relatively small amount of collagen deposition at that injury site. Again, which is pretty consistent with the robust regenerative response of young muscle. But when we perform the same muscle, these are just representative videos, but when we did the same imaging in the young mice that had received shRNA to Clotho, what we see is a markedly impaired regenerative response. Essentially, we see a disorganization of the muscle fibers. We see a lot of gaps, much fewer myofibers, which display this active regeneration, and again, collagen deposition throughout. Here in the cross section, I hope you can appreciate that there's a lot of spaces and a lack of organization. Once again, we can extract out the myofiber channel, leaving only the collagen, and essentially what we see is an abundance of collagen that really goes throughout that regenerating area. Very reminiscent of what we would expect with an aged skeletal muscle. And so here on the right side, we have quantification of this. When we look at the number of centrally nucleated fibers, indeed we see a significant decrease when we inhibited Clotho. We look at myofiber cross-sectional area, and just to make it very clear that this is myofiber cross-sectional area of the actively regenerating muscle fibers. And so we saw that the actively regenerating muscle fiber area was significantly smaller than non-targeting controls. However, it was also interesting to us, though we don't exactly understand why, that there were a number of these large misshapen myofibers also within that injury site, and these were not actively regenerating. So then this increase in local Clotho expression and seeing a connection between Clotho expression and the regenerative response next led us to turn our attention to muscle stem and progenitor cells to evaluate whether the progenitor cells themselves might be expressing Clotho. So we isolated muscle progenitor cells from young and aged muscle, and indeed we found, just as we saw at the whole tissue level, a dramatic decrease in Clotho intensity per cell of the aged cells when compared to young counterparts. And also we looked at Clotho secretion by the muscle progenitor cells just by performing an ELISA of conditioned media from young and aged MPCs. And what we see is that the young MPCs are indeed capable of secreting Clotho, but that secretion is significantly blunted in aged MPCs. So now some of the studies in our paper had evaluated then in these young and old MPCs, had evaluated Clotho's role in cellular senescence. And just as has been shown in other cells and tissues, Clotho seems to play a role in the attenuation of cellular senescence. So, and we saw the same thing in our muscle stem cells and our muscle progenitor cells. And so this was really intriguing to us, and it made us wonder whether mitochondrial dysfunction, commonly associated with aging and age-related tissue declines, may underlie some of Clotho's role in the attenuation of cellular senescence. And so we decided to evaluate mitochondrial function across our experimental cell populations. So this was a collaboration with Dr. Ben Van Houten, who's here at the University of Pittsburgh, and using a seahorse analysis, we evaluated the oxygen consumption rate of our young versus aged MPCs, and indeed we found a significant OCR in our aged MPCs when compared to young counterparts. We wondered if we could supplement aged MPCs with exogenous Clotho, whether we could enhance that oxygen consumption rate, and indeed we saw a significant improvement. And then when we inhibited Clotho expression in our young cells using an siRNA approach, we saw again that a loss of Clotho was concomitant with a significantly decreased OCR. So then next we wanted to take a look at the potential role for Clotho in the maintenance of mitochondrial DNA integrity. And so this is a collaboration with Laurie Sanders at Duke University. And using a PCR-based method then, what Laurie determined is that, again, perhaps not surprisingly, aged MPCs display a significantly higher number of mitochondrial DNA lesions when compared to and normalized to young counterpart levels. However, once again, just as we had seen an improvement in the bioenergetics with supplementation of Clotho to the aged MPCs, we see a significant decrease in the number of mitochondrial DNA lesions when aged MPCs are supplemented with exogenous Clotho. And conversely, we see that young cells exposed to siRNA inhibition of Clotho displayed a significantly increased number of mitochondrial DNA lesions as compared to young scramble controls. But then we were really interested in how might Clotho be affecting mitochondrial integrity. And so when we looked at this same model of young scramble versus young siRNA siRNA cells that had inhibited Clotho, these are transmission electron micrographs. What stuck out for us was that the mitochondrial integrity was markedly impaired with a loss of Clotho. And again, this is pretty consistent with an aging mitochondrial phenotype. That is, we saw the mitochondria were large and vacuolated, and through this TEM, it was very obvious that most of the mitochondria in the images displayed a compromised matrix integrity. We looked at this a little bit more closely by evaluating cardiolipin, which is a phospholipid that is primarily expressed in the inner mitochondrial membrane. Here, whereas we see a robust cardiolipin expression in our wild-type muscle progenitor cells, we see that cardiolipin is drastically reduced in cells isolated from our Clotho heterozygote mice. And again, this would be consistent with the loss of matrix integrity. So then we decided to evaluate whether potentially restoration of the mitochondrial ultrastructure, and for this we used SS31, which helps to inhibit cardiolipin peroxidation. And so we used 3 experimental groups for these studies. We used wild-type mice, our Clotho hets, and then Clotho hets that had been treated with SS31 for 2 weeks. These are, again, the transmission electron micrographs, and I don't have the quantification here, but essentially, though, the Clotho hets display a significant increase in the number of damaged mitochondria. As expected, SS31 largely restored mitochondrial integrity, as you can see here through these really nice mitochondria with very robust cristae morphology. And again, this is demonstrated here through the expression of cardiolipin. What was interesting is that treating mice with SS31 led to a dramatic decrease in the number of mitochondrial DNA lesions in cells isolated from the Clotho hets. And this was also associated with a significant increase in basal OCR levels in the Clotho hets. But of course, what we really cared about was whether a restoration of mitochondrial matrix integrity through SS31 would restore functional muscle regeneration in our Clotho hets. And so here, it looks like the labels are cut off, but here on the left, we have wild-type mice. In the middle are the Clotho hets, and here on the right are the Clotho hets that have been exposed to SS31. And when we look at the number of centrally nucleated fibers, again, as an indication of active regeneration, we see a significant increase when the hets are treated with SS31. We care about function, and so we use this hang test method, which essentially we invert the mice onto into a bucket, and we just essentially measure the amount of time that it takes for the mice to fall off of the grid. And when we do this, comparing the 3 groups of animals, what we saw is that the wild-type mice, after injury, are able to display a markedly increased hang time when compared to the Clotho hets. But that administration with SS31 significantly restored the functional capacity of the heterozygous mice. We then asked the question whether Clotho supplementation could enhance the regeneration of aged mice in vivo. And so we were really excited by the in vitro findings demonstrating that even administration and supplementation of recombinant Clotho could enhance the bioenergetics of our aged muscle stem cells. So we wondered whether this might translate to improved regeneration in vivo. So we actually started off, the first experiment that we did was to administer Clotho via osmotic pump delivery. And so here essentially we implanted the osmotic pumps 3 days prior to injury. And these osmotic pumps are gradually secreting Clotho into the animal. And in fact, we found that that translates to an increased local Clotho expression within the injury site of our aged animals. There, what we found was that there was indeed a significant increase in the number of actively regenerating muscle fibers. However, we didn't see any difference in the myofiber diameter of the regenerating fibers when aged mice were supplemented with Clotho via osmotic pump. Not only that, but when we looked at the images, what we saw was that though there were more regenerating muscle fibers, there almost seemed to be too many centrally nucleated muscle fibers. And so we didn't see that nice long train. Instead, we saw that the muscle stem cells seemed to be kind of running amok. And so we didn't think that this would translate to functional improvement. So we decided to go back and do a different paradigm, and that is to look at Clotho IP administration this time, but really focusing in on the temporal characteristics of the timing of administration. And so we decided to try two different groups, Clotho administration, 1 to 3 days post-injury, and these are all in aged mice. And then our second treatment group was Clotho, which was administered on days 3, 4, 5 after injury. We chose this second time point because the idea was to mimic that Clotho gene expression that we had seen in our young mice. And so essentially what we found was we looked here at myofiber cross-sectional area, and when we evaluated the cross-sectional area across the three groups, we see that administering Clotho specifically within that 3 to 5 days post-injury window led to significant increases in myofiber area when compared to the other two groups. And this seemed to be functionally relevant because as when we evaluated force production across the three experimental groups, in this case we used a in situ contractile testing paradigm. These are force frequency curves from aged animals, saline injected Clotho administration from 1 to 3 days, and then Clotho administration from 3 to 5 days post-injury. Here again, consistent with the histology, we found that Clotho expression at 3 to 5 days post-injury led to the most significant improvement in functional recovery after injury. So taken together then, the summary of the experiments that we performed, this is kind of the model that we're proposing. And that is that young muscle stem cells are exposed to considerably higher Clotho, both within their environment, and they themselves seem to also express higher levels of Clotho. And that this Clotho is important for inhibiting mitochondrial DNA damage, which drives a ROS production within the cell. And so as Clotho is present to inhibit mitochondrial DNA damage, it helps to preserve mitochondrial structural integrity, thereby allowing the cells to more effectively move into a phase of activation and participate in active muscle regeneration. However, with aging, we see a loss of Clotho in both within the microenvironment and within the cell itself, and that this is associated with an increased mitochondrial DNA damage, ROS accumulation, and overall loss of mitochondrial ultrastructure. And we hypothesize that this helps to drive cellular senescence and ultimately a markedly impaired muscle regeneration with aging. And so those are the main conclusions of our paper, although certainly as we've conducted these studies, they've only led to a number of other questions. And so we can continue to try and understand better some of these connections and understand better specifically the role for Clotho and muscle stem cell health and regenerative potential. So with that, I want to thank my wonderful lab. I feel very lucky to be able to come in to work with all of these individuals. And this was very much a collaborative effort. The first author on this paper is Amrita Sahu, who is a doctoral student in the lab and has been with us for a few years now. Of course, I also want to thank this project was, as I've tried to highlight throughout some of the experiments, was really a wonderful collaborative effort with investigators both throughout the University of Pittsburgh, but also externally at various institutions who have contributed to these studies in some way. And I want to thank you for your attention.
Okay, thank you. If you can, okay. Thanks, it's a pretty nice presentation. I have a couple of questions.
Okay.
This Clotho, like, how does it work? Do they act directly, like, for example, on the cell membrane, or is there any receptor, or is it at the genetic level? How does it work?
So we don't exactly understand, how Clotho, and those are really the questions that we want to start to tackle as a next step. Because though we can demonstrate that Clotho seems to be playing a role in mitochondrial ultrastructure, we don't really know how that's happening. And so that's a really important, I think, next step for us to better understand the mechanisms by which it's happening.
Okay. And when you had administered Clotho systemically, like IP injections, is there any blood has anything to like, does it blood cleaves this Clotho, because there are a lot of
Yeah.
proteases in the blood. So have you seen any effect like with the longer term or anything like that on the amount of Clotho you inject?
So it seems, what we mostly focused in on is the Clotho expression locally within the injury site. And in fact, we see that systemic administration does increase local Clotho expression. One thing that was interesting for us is that when we did the local inhibition of Clotho via shRNA, we found a significant decrease in circulating Clotho levels in the young mice. So suggesting that potentially some of the Clotho in the blood is coming from the muscle and the injured muscle.
And the last one is, have you looked at the gene expression of fibers or anything like that to look into like before injury, after injury? Because you mentioned about wind signaling, right? It blocks the wind signaling.
Right.
So have you looked at the gene expression profiles of these at different time points?
In the heterozygotes?
Like all the general gene expression, like just look at the profiles, like that is kind of a fancy thing these days everybody does.
Of course. No, so we haven't done that in the Clotho heads per se, although we have done it. We performed RNA-seq analysis on young injured muscle, old injured muscle, and then old injured muscle that has been that has received Clotho systemically, and so that's been revealing some interesting targets by which we think that maybe Clotho is having some of this impact on mitochondrial function. So I think it's definitely an area where we need to continue to explore further.
Okay, thank you so much. I really appreciate your time and effort. And your presentation would be on our website immediately as soon as we are done. So I'll send you the link also. So if you find some new things coming out in next several months or so, you can give us a call. We can do it again, something new. And thank you very much for your time and support for Science Mission.
Yep. Thank you for the invitation.
Thank you.